Good day. So I finally got the images from the graphic artist. That was a interesting experience. Okay, so moving forward, we'll do a little different. But I really like what I got, so I held on. So sorry, posting late. However, I think that the pictures were worth it. The images were worth it. So, with that said, we give thanks and let's continue. So I put together a very gentle explanation of the cell membrane. So I have these images that are going to show you some items that are actually our cell membrane for the cells. Now, the piece that I need us to remember is that different all of our cells have this membrane, and depending on where the cell is, you might have some different features included along the cell membrane that other cells may not have. But the general idea is what I'm going to present to us, because we're not going to, like I said, well, you didn't see it yet, so like I say in the beginning of the PowerPoint slide I've put together, is that it's a very gentle intro for seamless understanding, right? Because that's the whole reason for doing this channel, is to make sure that we're getting information that makes sense to us so that we can use this information and research more on our own, share with our providers, primary care and or specialists, and make decisions and inquiries about what we're going through, what's going on for us, in a more knowledgeable way to get the best results for our specific situation, with our mindset being what it is and respectful of what you hold important, and to marry that with the type of care and the remedies or solutions for taking care of whatever your situation may be. Okay, so I will explain all things as I do. And the first slide, you might have to press the speaker button, because I gave a little voice, a little media to the intro of the slide, and I wrote that down in the notes so that you can see it. This is my first time putting a PowerPoint on, so I hope it goes well. So what I think I will also do is do something in this section over here so that you're able to see it. I'm going to run it over and see, because remember, we're looking for seamless understanding. So enjoy the photos, the images. I mean, you might not get excited about them. I did, but you may not. Enjoy the explanation anyway, okay? All right, so let me know how it goes, what was clear. If something wasn't, let me know what that was so that we can do it over and get it right for you, okay? All right. Here is the gentle introduction to the cell membrane. So we're just going to go over the cell membrane as it really looks. These are the pieces that you're watching right now. This is our first slide, our gray slide, just to get us into what we're going to be looking at. So this is what most cells in our body look like, give or take some minor changes in the members inside of your layer there. Remember, in our first intro, before we got our picture done by Sing Studio, so we are grateful for them for that, I was showing you with my hands the phospholipid bilayer. So we now have everything. I have everything labeled for you. So this is the model that we're going to be looking at during this video. There will not be a lot of science jargon. I will only give you what you need, and you will be able to understand this just at the beginning. This is our intro to the cell membrane. Very basic, very seamless, very understandable. And this is, going to be our first. So some of the things that you see on the top, these are the things that you would see. These are true things that you would see, the lipids, you have your glycoproteins. These are the things you would see. But not everything we're going to cover. We're going to go to the filaments portion. So we're not going to touch that. We're not going to touch the surface protein, because once I talk to you about the proteins that are there, it's one thing. So it's a general introduction, so we don't need all the specifics, but I did want you to be able to see exactly what it is that's on our cell membrane. So welcome, and we shall begin. So this is our first slide. Now you see at the top, we have the extracellular fluid. That's the situation in all cells. That's a fluid medium that all cells exist in, because we have to be able to move. Now the top portion of this cell, you see things like carbohydrates just at the top, right? You have your carbohydrates. You see these branching circles that are coming out. You see something called a protein channel. You have something that's labeled glycoprotein, globular protein. You have your circles at the top. These are the heads of your phospholipid bilayer, the portion that likes water. Remember? We talked about that briefly in the other video, right? And then you have your carbohydrate chains that are branching out from these proteins. So these blue things that we're seeing, these are our proteins that are in our cells. Now just staying on the top. So remember the previous video I talked to you about there being a glycocalyx, right? It's that protective coat that's over the cell. Now the glycocalyx that you don't see right over, you see the extracellular fluid, it would be closer to your circles at the top, so that these pieces that you see, these circle streams, these chained carbs, these chained sugars, because that's really what they are, carbs break down into sugars. Sorry, not a lot of science, just what you need. I digress. This glycocalyx would be on the top, and that's to allow for cell recognition from other cells and to protect the cell surface. All right, so this is where we begin. So this is our phospholipid. This circles with those wavy lines. Those wavy lines there are your portions that are inside. Those two sticks, these are the fatty acid chains that we talked about, the ones that do not like water, the hydrophobic tails on the bottom up toward the this is your right. The right of this slide, or right of this image, I apologize, is going to be what the two look like. Remember, I was using my hands the last video with the circular head and the tails, the two tails. This is what it would look like, okay? And this is how this bilayer is actually set up. This is what it would look like. And the fluid exists in between. So now this phospholipid bilayer toward the top, right? At the top is going to be consisting of phosphatidylcholine, sphingomyelin. This is majority of what it's going to be at this top portion. Towards the inner portion, you're going to find, so in the inner portion on this nice gray slide we have is in the middle. So we have where the hydrophobic tails are, where the fatty acid tails, those nice little crinkly tails are. You're going to find your phosphatidyl ethanolamine, you're going to find your phosphatidylserine, and you're going to find your phosphatidylinositol. Now, why am I telling you this? Because all of these are key to cell signaling. Now, the last one, the ones that are inside the phosphatidylinositol, That's the one that's going to be key in a lot of cell signaling cascading events that are going to come up again when we start discussing maladies from a cellular point of view, that we can understand the cancers, the diabetes, the inflammation from the first video of our starting at the beginning. Right. So I want you to remember these things. This is a portion of our foundation. Now the glycolipids that we see they're going to be found in the outer portion of our cell membranes, and they have these carbohydrate chains that we see from in our picture. This is what we're actually looking at. This is what it would look like. Now, you see the yellow, well, no, it's gray. So you see these circles that are chained next to your proteins towards the inside of our phospholipid bilayer. That is our cholesterol, and cholesterol is important. It helps to keep the structure of our cell membrane, and it helps to keep the fluidity of the membrane, which is very key. So when the temperature within our cell is high, it makes for a less fluid cell membrane, which means it's going to reduce the number of small molecules that can come through. And when the temperature is low, it will then increase the number of small molecule permeability and make for a more sorry, make for a more fluid cell membrane layer. Now let's look at this. This is a good picture here. I like this picture a lot. So we see inside the scent, you see these you have the hydrophobic heads and hydrophic tails and hydrophilic heads within this phospholipid bilayer. If you will, you have to understand there is fluid all over. So you have the extracellular at the top, inside, in between, there's also some fluid, and this is where in between you also have interactions that we're going to talk about later on. Now let's come into the cell. We talked about already the cholesterol. They are there embedded within the cell membrane, again, for the fluidity purpose and for structure. Talked about that. Let's now talk about our proteins that we're seeing. Some of them don't need to be discussed, so we will talk about this protein channel. It's in parentheses. I had to put in parentheses for transport protein. Now here, this is going to be that U thing that I talked about before. This channel is being held in place by lipids. Okay, so very similar to the glycolipids that you see right there. They're actually also anchoring these protein channels into place within the cell membrane. And this cell channel is going to be this open pore, like I described to you before, but now as you can actually see in our photos, that allows passage of appropriate sized molecules that have the correct charge to cross right through onto the other side of the membrane and get inside the cell for things that are needed, like oxygen can get through, the waste from respiration, which is carbon dioxide, CO2, that can get through, things like of those nature. The protein channel opens and closes in response to the cell signals that come from the top, which allows for the control of movement of ions across the membrane, and this is going to happen seamlessly. There's no energy expenditure here for that to take place. Now let's look at this integral protein. Also says globular protein. The integral proteins are going to be, as you see, they're embedded within the phospholipid bilayer, and they span the entire bilayer. They span it. So all the pictures we've seen, if you take out a cell, you would see them throughout the span of your cell membrane. And some cells will have more, well, some cells could have less, but they're still going to span this cell membrane. And they'll have the portions that are exposed. So I'm putting my finger like you can see me, but the portions of your integral protein that are exposed either at the bottom or on the top are going to have a sugar coating over it. It's called glycos glycosylated. So it's the sugar coating. I don't know if I went it. I moved my face, so you might not have heard me. I'm going to say it again. So that if whatever is exposed on any of the proteins is going to be glycosylated. It's glycosylated. It's a sugar coating that goes over it to protect. And again, the glycosylation is always going to, like the glycocalyx, is always going to allow for protection of that piece of the cell and to allow other cells to recognize what it is on either side. So whether it's at the top or the bottom, this is always going to be the case. The next one we're going to look at is the peripheral proteins. So let me see what we have. We have this alpha helix protein here and surface protein, and we're going to talk about these together, right? So this is all the proteins is the same thing. They're going to, now peripheral proteins are going to be like the ones that are not embedded. They are not embedded. They are in the peripheral, on the peripheral of the other proteins. And they're there to help with protein-protein interaction. Again, when we talk more about signaling, we will have a lot more to say about them at that time. Now let's look at this nice big circle piece right here. Our globular proteins, the ones that look like they're just budding up. Let's talk about them a little bit. Our surface proteins. These globular proteins, surface proteins, globular proteins, they're right now doing the same types of things. Globular protein is a surface protein. Surface proteins are surface receptors, cell surface receptors. They are going to start off a chain of events, of intracellular events that are going to ultimately reach the cell's nucleus, which is the nucleus of a cell is, if you will, the brain of the cell. It tells what's going to happen, what to do, when to do it. So these surface proteins are going to act as a cell surface receptor and look out for its key piece. Remember when I showed you the 2 Lego blocks, talking about them fitting seamlessly and then going down? These are the things, this is another place where that takes place for this surface receptor, and it starts off this chain of events, reactions, that then reaches the nucleus of the cell, the targeted cell, and then results in the programming changes and the expressions that we then see of diabetes, in cancer, in inflammation, and things of that nature, in mobility of the cell. So what happens here with your surface proteins, the physiology begins to switch once the connection from the cell surface receptor, and once it has received what it is that it does receive, because everybody's specific, everyone has its job, it will start to change its form and then respond to a whole bunch of up and coming interactions, signaling exchanges that will take place. So this is it. This is our very brief. This alpha, again, I'll go over it again. The alpha helix protein is just another type of integral protein that's embedded. So both sides, the one that extends outward and the one that extends inside, are going to be glycosylated. They're going to be coated, and they're there to say, okay, this is what we do, and it's going to help facilitate signaling as necessary. Okay. All right. So as far as the filaments go for the cytoskeleton, that's just allowing for movement within the cell. And we can rest there because right at the time we don't need anything else. So I think that covers it. If there are any questions in regards to our small introduction to what our cell membranes look like, please give me that information. Tell me what you need, because our next video together is going to be talking about the cell signaling that happens from the beginning through, takes us right through the bilayer, our phospholipid bilayer that we see, and bring this into the cell and starts to show us the steps and what takes place. So I hope this was helpful. Thank you for listening and watching. Don't forget to subscribe to the channel and give me the thumbs up. and give me your comments. Be well until I see you again on Monday.